Today's a showcase for the lab. I had a whole series of lab coats ready for them, and everybody could wear a lab coat and goggles. And then for the setups today, we had a crayfish abdomen prep, which showed nerve recordings, and they could get the idea of recording from a single nerve. We had that where then they could record, they could actually move the preparation around and actually see the activity like that. So they actually could record from a nerve today, extracellular. And then we had Mr. Crayfish here, which we could talk about the crayfish, and we could tell them that they could shake hands with the crayfish. And we talk a little bit about why people use the crayfish as a model. And then we went to the Drosophila and the aspects of development in Drosophila and why people use them for a model in physiology and neurobiology. And then that they can see the Drosophila. We showed them a few genetic differences. And then we went over to start with some more physiology. Here's a heart rate recording in a crayfish. We actually had that where you can actually see the recording going along, and then we could stimulate the crayfish and you could actually see a change in the heart rate, and we could talk about how that happens. And then we had our favorite animal, which nobody wanted to feed, and that was our leech, and why people use leech. So that went over kind of. And then we talked about our mutant lines that we have in flies and how you get your F1 cross, for example. And then we went over here and we showed them a live prep that was here in the heart rate of a Drosophila and why people use the Drosophila to measure heart rates, and some preparations over here. And then we showed not only that about the heart rate and Drosophila, but then we had another prep over here where they could actually look at the neuromuscular junction of Drosophila, and you can kind of see it through the microscope there, and how you stimulate the nerve and record from identified muscles in a Drosophila larvae to look at synaptic transmission. And then we had behavioral assays that were over here where they could actually look at mouth hook movements of Drosophila and they could count them, and then we could do something where they would change. And then we showed them all the developmental different stages of Drosophila and body wall locomotion that's used for an assay as well for looking at effects of synaptic transmission or mutations. And then we had a preparation over here on the opener of the opener muscle, and then they could get an idea of why people use the opener muscle in the crayfish, because it's innervated by one excitatory motor neuron but lots of different muscle fibers. It makes it really easy to do some quantitative measures, and we had some traces going on there. And then also so they could see the opener muscle in the crayfish, we have one dissected here, and that why people have been using this preparation for years, like people at Harvard Neurobiology Medical School have been using this for quite a bit to understand synaptic transmission, and Bernard Katz, and first described in the anatomy of the opener muscle in 1888 by a German group with methylene blue staining. So we did a lot of preparations for everybody to see and understand what we're doing in the lab and why we do these things.